So my presentation is, I think, going to be more about reflections on progress over the years and the promise of cancer research going forward. And as was highlighted in the introduction, I've been at this for a while. And so I've seen a lot of dramatic changes, and I want to reflect on that. So my starting point is really to think back to, when I was a kid. I didn't do Irish dancing, but I was very interested in being active and sports. But, some of the things that I reflect on, television or telephones were devices that were attached to the wall. Probably nothing like anything anyone born in the last 20 years could imagine. The Stanley Cup belonged to Canadian teams. Again, kind of in the bottom left corner, that's a Toronto Maple Leaf standing in front of the Stanley Cup. And that's probably something that anyone born in the last 20 years may never have the opportunity to ever see again. And from my own perspective, I defined the seasons based on the sport that I was involved in at that particular time. And both of these pictures are actually, I'm in both of these pictures. You can guess which one I might be, but I was passionate about hockey. I was passionate about basketball, baseball, and anything else that any of my friends might get me involved in. But that was an era when we intuitively knew that cancer was bad from the perspective it made people very sick. It was also very mysterious. We didn't understand it and didn't talk about it. But at the time, there was also hope, because the impossible became possible. And around the time when I was still aspiring to play right wing for the Toronto Maple Leafs, or first base for what would become the Toronto Blue Jays, a couple of the events that caught everyone's attention, the moon landing was one, and then another one was a heart transplant. Actually being able to make a successful transplant of a life-saving surgery that right now is routine, but it was done for the first time when I was a teenager. So since then, there have been so many transformative changes. Instead of those telephones attached to the wall, we now have handheld devices that connect, entertain, and inform us. The Stanley Cup is a Canadian export, and I can't even think of the last time when I saw a Canadian team hoist the Stanley Cup, but hope springs eternal. And there have been tremendous advances impacting cancer. And what I'll be presenting sort of builds on themes that we've heard from a number of the earlier presentations. We have unimaginable technologies. There's been an explosion of information, and we've got examples of remarkable success. So cancer treatments based on knowledge. And we've already heard about all of these things from Allison, from John, from Colette. And so I'm going to go through some of these critical advances. So the unimaginable technologies include, computers and everything that comes with computers, the capacity to store information, the data analysis, and now artificial intelligence and machine learning. Robotics and automation. And so in a lot of respects, the robotics, we have to tell the robots what to do. but once they start working, they're exceptionally precise and they're tireless. Whereas if I was doing some of the work that the robot was doing, I'd be distracted when the telephone rang, or I'd get tired and lose my concentration. So these have been tremendous advances. We've got the emergence of these imaging technologies and scanners. So non-invasive detection of what isn't visible with our own eyes. Allison highlighted a number of these in her presentation. DNA sequencers and mass spectrometers for the fast, accurate identification of genes, gene products, and other cellular constituents. We also have instruments with the resolution and the precision that enable us to determine the three-dimensional structure of cellular components. So when Matt Maitland was talking about the central dogma and genes leading ultimately to proteins, we can get the high resolution structure of a protein at a level where we can see every single atom within that protein. And this has been a tremendous advance for the development of targeted drugs. We have microscopes with video cameras so that we can watch the dynamics and regulatory events as they occur in living cells. This is actually an image, and if I put my cursor, what that finger is pointing to is actually a cell that is undergoing an active form of cell death, and it shows a very dynamic process that wouldn't be evident to us if all we were doing is looking at the cells or taking photos as snapshots. So these unimaginable technologies have revealed so much new information in so many areas. And that has fueled this knowledge explosion. And so I think you've already heard of some of these numbers, but the human body comprised of 37 million cells and roughly 200 different types of cells. And these are really different factories. And I think Colette put it really well, talking about, the difference between a brain cell or a liver cell. So these are little factories that have to have exquisite control to ensure that they maintain our overall health and fitness. We've heard in a number of the earlier talks about the human genome. So that's the entire complement of genes or blueprints for every protein that's going to be expressed. And so the human genome has about 20,000 genes. And then the genome results in the production of many, many proteins that collectively we refer to the proteome. And the proteome is a dynamic complement of proteins. And so whereas the genome is relatively static and stable, every cell has essentially the same genome, every cell has a dynamic complement of proteins that is constantly changing during the different stages of its life cycle. And then different cells also differ from one another on the basis of the proteins they express. And we're now in a situation, so thinking back to what Lee presented in her introduction, we were all inspired by the promise of the human genome. And what she was describing was the Human Genome Project, which got started in the late 1980s and kind of reached a major milestone around the turn of the millennium. And that was the complete sequence of the human genome. And at the time, that was a singular sequence. So we knew in general terms the sequence of the human genome. Right now, we're in a situation where we now have hundreds of thousands of human genomes have now been sequenced. And that shifts the opportunity from just knowing the general landscape of the genetic makeup of a human to being able to look at what distinguishes individuals from one another. And the promise for cancer is to be able to look at the genetic signatures for many different forms of cancer. And so one of the resources that many of us use on an ongoing basis would be something like the Cancer Genome Atlas, which is really a catalog of the genome of individual tumors and individual cancer cells. And this has lent itself to the identification of cancer-causing mutations. And this was highlighted by Matt, where he was highlighting how mistakes in the blueprints for proteins could lead to dysregulation of regulatory events within cells. And so these proteins are the machines that do the work. They provide the structure that gives each individual cell or organ the structure. And they're also very important for the transmission of regulatory information. And so what you can see on the bottom left of the slide is really a depiction of, I think, what Matt has already shown, how these proteins become organized within regulatory circuits and networks. But that's actually tremendously oversimplified. And what's illustrated on the right is probably a more accurate depiction of how these proteins come together in these regulatory networks. And there's a tremendous amount of complexity, but also dynamic behavior. And so it's a real challenge to decipher what is the composition of these regulatory networks and how do these regulatory networks function with each other. But some of the other things that we've been able to learn from this explosion of knowledge, so going back to those cancer mutations and understanding where individual proteins are situated within these really complicated circuits, we can start to identify the short circuits. And one of the other tools we've got, and what's illustrated in the bottom right corner, is the high resolution structure. So this would be the structure of individual proteins at atomic level resolution. And in fact, this particular structure, which I would say would be in a ribbon diagram depiction, because it's really just illustrated as colored ribbons, that's actually a high resolution structure of a gene product that's mutated in cancer in a complex with the drug that neutralizes it. And so this gives us insights into how the drugs work, but also the types of drugs that need to be developed to neutralize or target these cancer-causing mutations. And there's publicly accessible databases where all of the information from these three-dimensional structures is deposited. And I looked this up this morning. There's now more than 170,000 different protein structures that are there in these publicly accessible databases that we use to guide and inform our studies. So Unimaginable Technologies an explosion of information, and this has lent itself to some tremendous successes. And, many, many successes, but, just highlighting one from the perspective that, this got a lot of attention. So Gleevec is a drug used for the treatment of chronic myelogenous leukemia. And here it is profiled on the cover of Time magazine. This is more than a decade ago, but it was the demonstration that if we understood the underlying mutations that led to the particular form of malignancy and had information about the structural implications of that mutation, it was going to be possible to design a targeted agent that could specifically neutralize or modulate the activity. And so in essence, the drug that is used to treat this form of leukemia selectively targets the cells with the mutant protein, has minimal effect on the cells that do not have the mutant protein. And so this takes away the selective advantage that enabled those leukemic cells to become harmful. John Lenahan's presentation. So John highlighted the promise of immunotherapy, and that's been another recent breakthrough by understanding how the immune system works to protect us from pathogens, but also how it can be reawakened to selectively target tumor cells, has lent itself to some absolutely spectacular successes. And everything that John highlighted suggests that there's many more successes on the way. So with the unimaginable technologies and the knowledge explosion, we can now begin to apply precision instead of intuition for the precise diagnosis and targeted or precise treatment of cancer. And so in our own lab, I'm really only going to highlight this in very general terms because we've capitalized on all of these technologies, the explosion of information, and we continue to aspire to move towards precision approaches to understand what we're dealing with. So as was highlighted in the introduction, I've got a background in chemistry and biochemistry and additional training in pharmacology and cell biology. And if I look at cancer cells, so these are electron microscopy images of normal cells on the left, cancer cells on the right. It's obvious that they look different. But the question, why are cancer cells different? So we've aspired to understand how they're different at the biochemical level with the expectation that this information would enable or instruct both the precise diagnosis of the cancer, but at the same time also more effective prevention, because if we know what the biological or the biochemical determinants that distinguished the cells from one another, we would know what to look for even before the cancer was detected. And then the secondary question, as we look to distinguish normal cells from cancer cells, is really to try and understand, what are the advantages that are gained by the cancer cells that enable them to become destructive, but at the same time, wherever they gain an advantage, they must also be exposing themselves to new vulnerabilities. And so when we think about precision targeted medicine, the goal is really to be able to neutralize the specific advantages that distinguish the cancer from the normal, and then exploit the vulnerabilities of the cancer cells so we can eliminate or neutralize the cancer cells without a major impact on the normal cells. And so over the years, our lab has really used a multidisciplinary approach. The present time, right now, we've got expertise in computational biology, proteomics and structural biology, which is really looking at the protein networks and how they're influenced in cancer cells. And we've got typically about 10 individuals in the lab, ranging from senior, very well trained and experienced scientists, all the way down to undergraduate research assistants. And within that mix is also 3 individuals doing their doctoral studies. But beyond our own lab, we also interact at a very high level with collaborators and other scientists locally, nationally, internationally, and a lot of sharing of materials, expertise, and knowledge. And where we need to, we establish the partnerships. And this is a theme that I think has been highlighted with all of the previous presentations. I'm not a clinician, so we need the connections with clinicians to inform the relevance and really provide, the materials that are real cancer cells or real tissues compared to just something we've established in the lab. And recently, we've had a lot of interactions with medicinal chemists, both in academia and in industry. And this is on a trajectory towards the development of drugs. And really, we're unified by our goals, despite the fact that we've all got different specialties. So when we look at how all of this comes together, when I look at the work that our lab is doing, our work tends to evolve kind of within this cycle where, we're identifying molecules that are implicated in cancer. We study the characteristics of these molecules at atomic level resolution, but we're really interested in determining how these individual molecules are situated within the network and how that can be transformed in cancer cells. But then how can we turn that around to now manipulate those molecules to modulate that network, really to neutralize the advantages and exploit the vulnerabilities of the cancer. So this really is all directed towards identifying the underlying determinants of disease at the individual level of molecules, but then really being able to develop targeted interventions using targeted agents that are precisely targeting those molecules that are perturbed in cancer. So my very last slide is really just kind of a summary to pull all of this together. So if I reflect on where we've been, what we're doing, and where we're going, the technologies that we're working with continually get more sophisticated, and it illustrates that the cellular networks, the biological networks we're studying continually get more complex. But all along, the questions that we've been addressing and our goals have remained the same and are relatively simple. We do this as a collaboration and through teamwork within our own lab and beyond. And so what brings us together is our shared aspirations, and we're really motivated by our aspirations, not our tools. So again, I want to thank everyone for the opportunity to participate in this event and for your attention.